Welcome back to Physiology Raw. This is essay questions for exam 3. This is the first question. My last video was way too long, so in an attempt to make it shorter, I've drawn a few things before starting. Alright, so right here's a graph of the ventricular muscle cell action potential, and then this is the SA node or the autorhythmic muscle cells. And color-coded a little bit, so let's get right into it. The question is asking to describe the action potentials in the cardiac ventricular muscle and the SA node, and then describe each phase of the action potential. Describe the channels, ion currents, and membrane potential involved. So this red region is called phase 4. And that is characterized by sitting, well, in this case right here, it's sitting closer to negative 90 millivolts, and that's because in a ventricular muscle cell, it's more permeable to K plus, because there's more of these IK1 channels. So the I represents I equals current. I equals current. So there's more of these K channels, making the muscle cell sit closer to the Nernst value for potassium. And so first I'm going to run through this one, and then I'll relate it back to the other one. So this next phase, it's going to be called, you see that all right? Ooh, that yellow. Phase 0. And that is characterized by the depolarization. You can see that it shoots way up. So, depolarized, and it does this by opening sodium channels, or the I and A channels. And so that's going to quickly depolarize the cell, and also is closing. So this is closing the IK1 channels. Very good. So this next part, so we hit the peak, and you can see that there's a little bit of a slope, so we're going to get right into that. This peak, or just after the peak, is called phase 1, and that is characterized by the rapid repolarization, and then that is done with, so the NA channel closes, I and A closes, and then a small number of potassium channels open up, along with chlorine or chloride channels. And so those are the I2, 1 and 2. So this is the 1, that's the 2. And so the potassium will go back out of the cell to bring down the charge, and chlorine will rush into the cell to bring down that charge as well. You can see that it levels out a little bit, and we'll get into that. That is called phase 2. So that's the plateau. Plateau, I spell that plateau. And so this is because voltage-gated, V-gated calcium channels open up as well as sodium channels, but the sodium, or the calcium channels open first, and then there's sodium channels that open up as well. And so that helps. Then there's 2 types of the calcium channels. There's the C type, which is long-lasting. So long-lasting, and then there's also these T type, which is a transient. So they don't last as long. So those are a little bit shorter. And then there's also, so the reason why it goes back down is there's these the IKS channel, which is a sodium channel, right? Or not a sodium, sorry, potassium channel, IKR, then IKUR. Okay, so as you can see, there's 3 potassium channels. This one's and they're both characterized a little differently. So this is characterized by the inactivate, and then the activate. So these are long lasting because it takes a little bit for them to inactivate, and then these ones are a little bit faster in inactivating, and then these ones are reversed. So this one activates quickly and then activates more slowly, and because these 3 potassium channels, well, the reason why it plateaus for a little bit is because these channels negate what these channels do for a little bit, but then after a while the calcium channels start closing up and the sodium channel starts closing up, and then the potassium channels overtake and help curve the charge to go a little bit further down. Before I run out of space, do the last phase, and that's classic repolarization. So even more K channels open, specifically the even more of the IKR and then the IKS, and then finally back to the beginning, the IK1 channel opens back up. So these are opening. Open these ones, and then closing finally are going to be the last of the calcium channels. So I use this one as kind of a reference for each one of these phases. And so we can move over to the SA node. So at the beginning of the SA node, the IK1 channel is open, but it's getting depolarized by sodium. And once it hits its threshold, it'll shoot into phase 0 where the sodium channels will open up, let the cell get super positive, and it closes the potassium channels up. And then it runs over into phase 3, so just repolarization. Let me go back a little bit. I'll just use blue for now. But there's a kind of a slow plateau right here because the calcium channels close slowly. CA channels close slowly. So you see this kind of like instead of a sharp peak, it's more of a dull peak. And then finally, these ones, so on this region, potassium channels open back up, causing it to repolarize, and then we hit back down here to get our back down to our resting potential. And notice this one is a graded or a gradual depolarization. And also notice that there is no hyperpolarization. So these cells aren't designed to be hyperpolarized. Once they get a new signal, they want to be able to react to it because it's the heart. There's no point in having hyperpolarization. Okay, I think that covers that. So in part 2, we're going to talk about EK or ECGs, I guess. Let me erase this real quick. So ECGs are all about tracking that membrane potential that we just talked about a little bit. Get that recentered. So we have nodes, and then let's make it simple. Oops. That kind of works, right? No, it doesn't. Yes, we got the heart. The heart. Oops, alright. Alright, so the heart, and how we follow these, is that there's a bunch of different leads that we could play with. So the lead is just meaning where you put the negative and the positive nodes, I guess you could call it. And those will result in different waves. The second part of the question asks for P, Q, R, S, and T waves. So I guess we're going to start out with a P wave. So P waves start out because the SA node right here is going to send out action potentials. I wonder if I could, I might try to do this all on the same model, so we'll see if it works good. So because it's starting a negative action potential, so like the outside of the cell is becoming more negative, So when it starts out, inside of the cell is negative, the outside's positive, comparably. And so when action potentials happen, negative charges go outside of the cell. And so when a negative charge goes towards it goes towards the positive end, we see an upstroke. And then if, that's true, if we see a positive charge goes toward the negative, that's an upstroke. But then if we see like a positive charge goes towards the positive, that's an inverted stroke. That comes in a little bit later. Okay, so the P wave is the atrial depolarization, and so that's going to shoot out that way. When that is all good and done, we move over to the AV node, which is right about here, and so that's going to start polarizing. Depolarizing? Yeah, polarizing the ventricles. So it's going to shoot that way. We'll write that. I think it's going to be Q wave. And then, so once that's, done, a little bit that way. And then, after the Q wave is done, we have the R wave, which, trying to do different colors. R wave, right, which shoots down the ventricle. So now polarization is going down a little bit more towards the. Let me put this more accurately. So again, a negative wave is going towards the positive end, and then we have the S. So this is going to propagate all this way, and then up that way, and then the last place to get polarized is this section right over here, which is going up now. So maybe I do a different color for that, even red, and that is the S wave. So right here, they're usually, these ones are really tight together, so it's a QRS complex. And then finally we have the depolarization, or the repolarization, I don't want to have the, of the ventricle. So now all this stuff that was polarized is going to get depolarized. It's going to be the T wave. And yeah, so it's so I wonder if it would be helpful if I drew out a little something. So okay, so the S wave, that's going to go into our little Q wave, which was green, which is down, and then our big R wave, which is up like that, and then our little S wave, that, oh wait, I think this is all the way down, then a little S wave that shoots back up, and then to finish it off with our little T wave. Alright, I think that it's gonna have to do it. Yeah, so when is that everything it asks? So yeah, that's the flow of the charges that go through the heart, and the charges determine the waves. And the waves are kind of determined by the orientation of these two nodes, and that's pretty much how that goes. Hello, this is the question 2, essay question 2 for exam 3. As I said before, to make my videos go a little quicker, I took the liberty of drawing a few pictures beforehand to help me speed up the process. So this says describe the events taking place in the left ventricle and aorta. So I've drawn the left ventricle, aorta, and even the left atrium. So let's just get into it. So first, this is connected to the lungs, and blood is just passively falling into this, and some 80% of the blood passively falls into here as well, of the volume that is able to be in the ventricle. So this is a graph of the ventricle pressure and atrial pressure, and this is the volume of the atria, or the ventricle. And so I guess the first part, so once this hits 80%, just about, the atrium will contract, and so that's our little first bump of pressure right here. It's the contraction, and this contraction brings up the rest, this 80% to the full 100%, right? Very nice. And so now what happens, since that contracted, the ventricle will start to contract. So as this squeezes closed, this valve that was open, let me get rid of that. So this valve was open for a little bit. Since this pressure is climbing, it's going to actually snap this closed. So the pressure is going to snap the mitral valve closed, and this is the oh, come on, the lub sound of it snapping closed, because this ventricle is starting to contract, and that's what this spike in pressure is for the ventricle. And so once that happens, once pressure is high enough, once pressure is higher in the ventricle than it is in the aorta, this semilunar valve will open, letting the blood flow up into the aorta. And so Yes. And so notice there is going to be a second where this is contracting. And the pressure on the atrium is going to be enough where it closes, but it's not going to quite reach high enough pressure to open this valve yet. So there's going to be a little, was it isovolumic contraction. That's worth noting. Yeah, but once this does open up, it's going to contract, push everything out. So I guess I'll draw it like open like this for a second. But then once the ventricle starts to get too low in blood, then the pressure in the aorta is going to come back, and this valve will snap shut. And this is going to be that dub sound, because that's snapping shut. Include the various stages, blood pressure, opening, including the valves, heart sounds. Finally, be sure to explain the pressure volume curve for a single cardiac cycle. I kind of did that already, but I'll run through it again real quick. So this is the blood or the pressure volume curves. Yeah, pressure volume curve. There's a little bit of a note. Yeah, so the pressure will shoot, or the volume will shoot up for a second right when the ventricle contracts, shooting that in. So that's that little jump. And then there is a second of isovolumic contraction. Oh, actually, this is the first one up here. It says first, second. So the first one where it's contracting. This has enough pressure to close. This doesn't have enough to open. It finally opens. It contracts, pushes everything out. The volume drops. Pressure's still really high. Volume drops. And then there's a second when it's still contracting, but now the volume or the pressure in the aorta is high enough where this closes, but the pressure in the ventricle isn't high, or the atrium isn't high enough for this to open quite yet. So there's a second isovolumic contraction. This is contracted, nothing's leaving, both doors are closed, and then this starts to relax, and then blood is able to passively flow from the atrium into the ventricle once more. I think that covers that question. One last thing, just to be clear, these doors close because blood is attempting to go in the wrong direction. So high pressure in the ventricle closes this valve because there's low pressure here. So blood pushing it catches the flaps of the valve to close it, and the same concept works for this one. High pressure here pushes back against the valve which closes it. Alright, this is question 3 for the essay questions for exam 3. I'm going to do it a little bit backwards. So I'm going to talk about calcium in the cardiac muscles, including the mechanisms of its release into the cytosol and return to the SR and extracellular fluid. I drew a little picture of the cell to try to get things going a little bit quicker. So I think the first thing we need to point out is this channel. It's called the F, like the funny channel. And so what it does is it'll allow positive ions to go into the cell to make sure it never hyperpolarizes. We never see hyperpolarization in the cardiac cells. Okay, so with this, so unrelated to that, we have a sphere of influence or an action potential running down the cell. And notice it is similar to cardiac cells, but these ones have a much shallower and broader T-tubule, and this is to help help it never get a potassium buildup in here, so there's never like spastic contractions like you see in skeletal muscle cells sometimes. And so what that does is it'll open up voltage gated calcium channels. Let's see, gated calcium channels, and there's two types. There's a transient type and the L type. And so all that means is the T type will open and close quicker, and the L type takes a little bit longer, sustains the contraction a little bit longer. So calcium is red. It's going to run into the cell, and so there is a little bit of extracellular calcium that will cause the tropomyosin, actin-myosin filaments to contract, but the most of the calcium that we are looking for is in the SR. So and this is a ligand gated channel. So calcium from the outside will bind to that channel to allow these guys to run out and do their thing with bind to the troponin myosin complex. And there is a right here. So when the signal is given, there is a calcium pump back into the sarcoplasmic reticulum, SR, right here. And then there's also a pump right here that will pump out the calcium in exchange for sodiums. Na sodiums will come back in, and of course this is just representing the Na plus sodium potassium ATPase pump. So any sodium that is pumped in using that antiport will be pumped back out through the sodium potassium ATPase. And so that is how what does it say? Calcium is used in the muscle, or in the cardiac muscle, and how all that goes. So now we're gonna talk about the first part of that same question, which is the mechanism of inotropy, lusitropy, and chronotropic actions of norepinephrine and acetylcholine. So here we go. Let's do that. Yeah, so everything starts out in the same way, so I'll just make it black. So what is it called? The epinephrine or norepinephrine will bind to beta 1 receptors, and then that will activate the subunit A. Oh, the subunit A right there. And so this one has the ability to, I don't want to run out of room, so I'll just go over here and do it. Can you see that still? Yeah. And so right off the bat, that subunit can interact with L type. Remember the L type channels I was talking about over here? Calcium channels. And then that will increase calcium concentration in the cell, which could open the SR. So like activate SR. I should have done it in red. It's alright. Maybe I'll do the rest in red. And so that will increase contraction, which is inotropic effect. And so I guess it'd be worthwhile defining a couple things real quick. Maybe I'll just do it right here. So inotropy, it has to do with the strength of contraction. And so if we add more calcium, so it's pretty much tied with calcium. So if you had a bunch of calcium in there, then the contraction will be stronger. And then the lucitropy is the ability to relax. So like relaxing strength, I guess you could say. So if we make calcium shoot out, if we make it leave really fast, then this will go up. The lucitropy will kick in a lot quicker. Okay, so continuing on this way, the subunit A could also hang out with activate adenal cyclase, which makes cyclic AMP. So that concentration will go up, and then that will activate protein kinase A. So hopefully I'll have enough room. If not, I'll start erasing stuff. So I think since I started with the inotropy, I think I'm gonna just keep going with that, and then I'll erase and then put the lucitropy pathways. Okay, so this one, protein kinase A, can phosphorylate, again the L type, CA, oh geez, why am I doing like that? Somehow. L, so that's L type calcium channels, which will, increase the calcium concentration in the cell, and then that will that increase will then interact with the SR calcium channel as well, that ligand, and so like SR channel open, and then that will in turn increase inotropy because it's a harder contraction. And then the other one is it'll phosphorylate the RYR receptor, I guess RYR, which then I guess it's pretty much the same path, I guess you could say, but it takes a slightly different way. It messes with so like in that receptor there's a compound on it, and it's called, if I remember right, calstubin 2. Oops, sorry, that's calstubin 2. So I'm just trying to go fast. So that's going to open up that receptor, and then it's going to increase concentration. Wait, let me write that a little better. And so this will then, increase inotropy because there's more calcium in the cell. So let me get my Put eraser here. Okay, so this protein kinase A can also activate I guess phosphorylate, something called phospholambin, which is connected to that SERCA pump, which is, yeah, maybe let me write it right here. So that'll open up that channel, which will force it to reuptake of calcium from the inside of the cell. And so if that's pulling very hard, then obviously calcium concentrations will reduce, which leads to that lusitropy. I don't know if I'm saying that right. Or you could have that protein kinase A directly interact with tropomyosin actin complex. So let's go phosphorylate troponin 1, which knocks off that calcium, and then that will increase speed of relaxation, which is again the lusitropy effect. So if you remember from, I think it was like first unit or whatever, or maybe last one, we were talking about the skeletal muscle, and how troponin is one of the parts of this complex that if it doesn't have the calcium on it, then the myosin and actin won't be able to, the troponin and myosin won't be able to bind to each other. So that is the lusitropy effect. So let me move on over to the, where is it? Oh, acetylcholine. So this is right here. I'll just write it out. Neuro or epinephrine. So norepinephrine or epinephrine will bind to the beta-1 receptors. So let's move on a little bit. Does it talk about that? On the heart. So both at beta-1 that I just erased, so when things bind to the beta-1, that's going to increase heart rate. Yeah, so that's just going to increase the heart rate on that. So now let's move over to the what acetylcholine does. Choline will bind to the M2 receptor on the heart. And so let's see, there's a couple ways that this can go. There we go. And it's gonna slow HR. I'm gonna stop using that 'cause it's running out. Make sure my thing doesn't die. So there's the this pathway talks about opening this GERK channels, and that's like G protein. It's like rectifies. This is potassium. It rectifies the potassium. Oh, it's an active. So G protein active inward rectifying K channels. So that means it's gonna it's gonna make the membrane potential more negative because it's taking in more, or I mean, it's opening up potassium channels that wants to leave the cell. So if the membrane potential is more negative, then it's going to be harder to make a contraction. So heart rate goes down. And then it can also go the alpha subunit path. So activates the alpha subunit, which then decreases cyclic AMP, which this one will decrease that funny channel. So if no positive ions are able to get inside to maintain, so to make sure that the cell doesn't go into hyperpolarization, that means heart rate will also decrease, because it's basically like this one, making the membrane more the membrane potential more negative. So there's that. And then also on this, so from here, I guess I don't really need this one. In I channels, the calcium channel, influx of, or conductance of calcium will become more positive, meaning when this action potential comes down, it will have to be like a greater action potential to trigger these calcium channels. And so if those calcium channels can't open up, heart rate will then decrease again. Well, not again, but So it says, I think that's gonna do it for that one. Hopefully that made sense. Alright, this is question 4 for the essay questions. We're gonna be talking about osmotic or capillary exchange. And so right here I got the arterial end, and this one comes with a some smooth muscle right here that helps constrict and dilate, and that will help control some pressure, like some pressures. And this one doesn't. This one's the venous end. Venous end. All right, so the first main pressure that we see in both sides, but I'm going to start with the arterial side, is the hydrostatic pressure. This is 30 millimeters of 30 millimeters going out of the arteriole. And then the second one is it's called B cups. This is a blood colloid, which means protein osmotic pressure, and that is at 20 millimeters of Hg going into so like this helps retain water. Oh, I was not supposed to use that color. I didn't want to use that color. I'm going to make that yellow instead. So it's 28 millimeters of mercury going into the cell called B cup. Okay, and then there is the I cup, which is interstitial colloid osmotic pressure, so the protein's on the interstitial side, and then this is pulling water out, and it has a value of 8 millimeters of that per second. And then if you notice, this little green tube is the lymphatic system, and so that pulls 3 millimeters of mercury just out of the interstitial area, but also in turn out of the artery itself. So I will say that some 90% of water lost from the arterial side will return to the venous side, like it'll go back into the venous side, but on both sides of the lymphatic system that pulls out that remaining 10%. So on the venous side, we have the same pressure, so we have the hydrostatic pressure pushing out, but that one is only at 10 millimeters of mercury this time. Then we have that same amount of B cup, that 28 millimeters of mercury going out. Same for the I cup, 8 millimeters of mercury out, and then can't forget the lymphatic system with the 3 millimeters of mercury being pulled out of the system entirely. And so in the end, we have a total net flow of water of 13 millimeters of mercury leaving the arterial side, and then a -7. So this is negative because it's saying it's going back into the vein, into the vein. Okay. So that says, just reading the question, making sure I got all those parts. All right, yeah, I think I walked through it there. I drew something beforehand to try to make the video go a little faster, so I'll just keep on trucking. It says use an example of inflammation to describe the mechanism for edema. So edema is when there's a concentration of water outside of the vein or artery, so like in the interstitial space. So inflammation is caused from a histamine buildup. Histamines. This causes endothelial retraction. Retraction. So something I neglected to say at the beginning. So the arteries and the veins are lined with endothelial cells, and so those are just the cells that like separate, the interstitial from the blood, and so small particles like oxygen and carbon dioxide are able to freely go through, like right through the cells, but then the cells have a little space in between them for things like water, amino acids. What is that? The glycogen? No. Glucose, that is. So glucose, and they have to pass in between the cells. So histamine will cause those endothelial cells to get smaller so water will freely pass through, like in between the cells. It creates leaky uh, endothelial barrier. So let's just say like leakiness, and then that is caused the edema or the buildup of water in the interstitial space. All right, I think that covers the first part. The second part, I might actually pause it so I could set up for the second part. All right, so I just paused to erase and do a little bit of setup for the next one. Part 2 of the essay, describe the baroreceptors, include all the components of the reflex, and describe the chain of events that would occur if... Okay, so the baroreceptors are located on the carotid artery, and also on the aortic arch. And so they're stretch receptors. And so when blood pressure or blood volume increases, that means the baroreceptors would cause more action potentials, and that will want to reduce. So let's say, so if a stretch goes up, it would want to decrease. So I think it's important to actually explain this before I get into that, and it'll help me. So MAP is mean arterial pressure. So stretch would mean an increase in MAP, which would want to decrease either cardiac output or was it peripheral resistance, and I'll get into those in a second. And then if it's not being stretched enough, it would want to increase these two things to sustain that mean arterial pressure. And so yeah, so I'm going to talk about cardiac output for a second. Cardiac output is heart rate times stroke volume. And so usually when we want to change cardiac output, we're going to raise the heart rate. And then peripheral resistance is a longer equation, so it's 8 times this is viscosity and then length over pi r to the fourth. And since this r to the fourth, it's the most powerful thing that we could change. So this is a vasoconstriction or vasodilation. We can't really change the viscosity of our blood or change how far our blood has to travel. So we're going to change the radius of the arterioles. And so back to the baroreceptors. So they could either affect the heart rate, increase the heart rate, or change the diameter of some of those arterioles with constriction or dilation. So in the example of, what was it? Blood pressure suddenly dropping. So let's go BP drop. That means these are not getting stretched at all. So like no stretch in baroreceptors, which is signaling that the mean arterial pressure has gone down. And so what we want to do in response to that is we could either increase heart rate. I guess we could increase stroke volume by I don't know, increasing like inotropy, and then I guess increasing lucitropy a little bit, because lucitropy is like the ability to like repeat a stroke again. So, but increased heart rate is the main way we'd want to go about that. And also decrease, or sorry. So increase our peripheral resistance, right? Because we want that to go up. Okay, and so we do that with vasodilation. So if we make some arterioles bigger, then more blood can move through, and there's less resistance for the blood flow. All right, let me get rid of some of this for the last part, which is explain each of the following is involved in regulation of blood flow. Okay, so the regulation of blood flow includes, let me write these out for us. Generic auto-regulation. Let me just erase some more. And then we have, and then it says the Perkins, including their role in active and reactive hyperemia. So that should be fun. And so myogeneric regulation, as you can see, myo, so it's going to be some blood, and then the auto regulation suggests that it does it by itself. And so this is when, so smooth muscle has a cool system. So when you stretch smooth muscle, it automatically retracts. So like a stretch. It has some mechanically gated calcium channels. So when it's stretched, then it'll open up, and then the smooth muscle contracts. So contraction. Okay, and paracrines, it's going to be a little bit different. Paracrines including, so they come from the endothelial cells that we were talking about earlier. Endothelial cells, and they're going to release nitric oxide, and they respond to reduced O2 and increased CO2 levels. So this is usually from metabolic processes. And then we have some combinations of a few things. So adenosine plus nitric oxide is going to cause vasodilation. And then you have a few other examples that mostly causes vasodilation. Okay, so it was specifically, I think it'd be more important to talk about specifically the active hyperemia, and then the anemia. It's kind of sporadic. And then the reactive hyperemia, but we're going to start with active. So this is when increased metabolic processes are happening. So like, dang dude, I'm going to use a different marker, I guess. Metabolic processes, and as a result of that, like I said earlier, so we're going to see an increase in CO2 and a decrease in O2, and so that's going to make epithelial cells release that nitrous oxide release, and that will increase blood flow through that area. I'm gonna erase this one. So now we could talk about the reactive hyperemia. And so this is usually caused by some kind of clot or damage. So blood flow stops going to a certain area, and the epithelial cells will start releasing some, nitrous oxide, but then that more potent adenosine. And so both of these will cause crazy vasodilation. And so when blood flow finally is restored to that area, we'll have like the max dilation in that area, and so blood will, you know, re-enter that area, and we'll see some hyperemia through there. So we'll say no flow, a lot of flow, and so this is metabolic rates mostly. All right, is that the last of that question? I think it is. All right, I think that's all I got to say about that one. All right, this is question 5. It's going to be split up into 2 parts. So here's the first part. Talks about composition of plasma. So I drew a little diagram to help me get going quick. So this first part is the components of blood. So the first part is plasma, and that takes up about 55% of the blood volume. So plasma is colloids, or which is proteins. Proteins. The biggest amount of the protein in the highest concentration is albumin, which is makes about 60%. Dude, really? I'll have to change that. Another one. 60% of and it's more okay. 60% of the plasma is albumin, and that is responsible for the blood colloid osmotic pressure, which we talked about earlier in an earlier question. And then it is followed by the globulin component, globulin, right? And that is the antibodies, or I mean, these carry something called antibodies, which I think we'll get into later, but part of that is the AB blood type, positive or negative, or 0, or just A, or just B. So, and then the last part of plasma is fibrinogen, which is a clotting factor, which we'll talk about in the second part, but that's not quite where we want to go. All right, so, and then this is the actual like red blood cells, and then this little part right here is it's called Buffy's layer, which is the white blood cells and other leukocytes. And so a little bit about the red blood cells. If it is below that 45%, then that is equal to being anemic. And if it is above, that means you're probably blood doping. If you're above that blood doping. So pretty interesting right there. Now it's going to talk about especially explain how the heme is broken down. So we're talking about how blood leaves the system. Okay, just making sure you can see good. And so that is started with, so the blood is first taken to the spleen. Spleen. There it is broken down a little bit, and we want to talk about heme in particular. So, you know, the most of the red blood cell is just proteins and, you know, easy stuff to take apart. But heme is the good stuff. It has iron in it, and that's how it pulls oxygen around the body. So there's these macrophages in the spleen, that convert heme by taking apart certain bonds or undoing double bonds to make it a single bond, and so it converts it into Bill. Oops, that's not how you spell it. Blip, blip. Billy Verdon. Okay, Billy Verdon. And then that gets converted to Billy Rubin, which is the good stuff. Billy Rubin is taken by albumin. Albumin over here, it carries different things throughout the body. It's a little transporter. So Billy Rubin is taken by to the liver, where the liver puts a type of sugar on there. So I'm going to say it puts, let's say sugar on there. And then it could either go to, so there's like this unconjugated Billy Rubin. And then after the liver, it is now conjugated. And so all this gets secreted out into the bile, right? And so the bile in the intestines gets pushed like through the intestines until this bacteria in there eat that sugar that was put onto the bilirubin. And then this then converts that into the what was it called? Bilinogen. And so part of this can go into, right, especially, so part of this can be converted to a brown coloration, brown pigment, and that's for feces. And the other part, it could either go back to the liver and start over again. Oh wait, sorry, I skipped a step. So it could either be reabsorbed in the intestine and go back into the blood, where it could go back to the liver and restart, or it could go into the kidneys. And then this urobilin is then released into the urine. Has a yellow color. Okay, so that, okay, so that's part one, and I'm going to erase this and start part two. This is part two of exam question 5. So we're going to be talking about hemostasis and the three mechanisms that play into that. Hemostasis. And so in a second we'll get to this. But the first is vasoconstriction. And so when trauma or damage happens, the smooth muscle in the arterioles will constrict, and sometimes that will even completely shut off the blood flow to that area. But that's only for a short time. Eventually, blood will have to continue flowing into that, and that's what this next part is. And so we're going to go call that platelet platelet aggregation. So the first part of platelet aggregation, you see that there's these little bumps along the vessel, and those are the endothelial cells. So they will release, when they're damaged, they're going to start releasing this Van Willebrand factor. And so that, and also these blue things are the collagen, just sitting behind the surface of the vessel. And so collagen has a negative charge on those proteins, and so that will make the Van Willebrand factor receptor on the platelets connect to it. And so when that happens, and so part of that is when this receptor is bound, it'll start releasing different granules. And so I think first we're going to start out with these purple ones. Oh, that's blue. So let's start with these purple ones. These are called the dense granules. And this releases ADP and also calcium. Calcium, once there's a few of these platelets that have connected with the Van Willebrand factor, calcium will actually make it, because there's actin myosin filaments in there, so it can actually start contracting. And then ADP is used to activate other platelets. There's also these fibrinogen receptors right here. Actually, I'm going to draw another one right here. And that will come into play in just a second. And then these are alpha granules, and they will release factor 5 and then cofactor. So these are cofactors, and we will talk more about these when we get to coagulation, which isn't quite yet. And so, geez. So yeah, I'll erase this for now, but no, that's what those are for. Okay, so as Van Willebrand factor starts attaching to other platelets, we'll see that part of platelet aggregation is that these will start forming little ropes to other platelets. Let's try that one. And so each one has all those same components with the dense granules and the alpha granules. Just draw maybe two more to get the idea across. And yeah, so part of this, so that is the platelet aggregation, that once this Van Willebrand factor is out and the collagen activates one of these platelets, other platelets will come along and they'll start creating a net. And this purple is fibrinogen, which eventually will create the fibrin ropes. And then that helps tie everything together. And there's a platelet releases this growth factor from, I think it's the dense granules. So that is part of the platelet aggregation. And then, so like I said earlier, this this alpha granule will start the coagulation cascade. So let me erase some of this so I have room to talk about that. I'll make a note. Cascade. And so we'll start with the intrinsic pathway. There's a variety of, there's two different pathways that it could take, and they both lead to the common pathway, which is the second part of the question. So the intrinsic. Pathway. And so that starts out with this factor 7, which turns into activated factor 7. And that is so this is a plasma protein that is from the liver. And then when it comes into contact with this negative charges right here, it will become activated. And so that's just one of the first steps to start going. And then so an activated version of this will activate the is that 9? And so 9 is from yes, let's see, thromboplast, right? Thromboplastin, which from the name we could see that it talks about this thrombonin, and it's it's an accedent to thrombin, which is necessary for coagulation. So activated that will go to activate factor 11. So activate factor 11. Oh. And then that activates what we need, factor 10. Oh, this one actually goes with that one that the alpha granule produces, factor 8. And then I think it's factor 5. And so those two together, and so this is active plus factor 5. And then that activates. So now I guess I should draw a line right about here, right here. This is the common pathway. Let me just write these down and then I'll run through them. This is prothrombin, creating that thrombin, activated thrombin, activates fibrinogen, and then so that's fibrin right here. And then for the sake of just keep moving, we'll do the extrinsic right here, and then I'll run through what they mean. So that's going to be active. So there's this. Let me give me some more room. Jeez. So there's tissue factor 3. Factor. And finally back to, oh yeah. X A plus V. Okay. So going back to this, there's a thromboplastin, and then that's going to activate the 9th factor, which is an antecedent to thromboplastin. Yeah. And then that activates number 10, which is just the start of the start of the common pathway. And so what all that does is it allows prothrombin to change to thrombin, and then once that activated, then fibrinogen can change to fibrin, which is the stuff that holds all the platelets together. And so this one over here is the extrinsic pathway, and it starts out with this factor 3, which is tissue factor. So if something outside of the blood gets in, like proteins from the collagen, it'll start this pathway. Starts with factor 7, which again is another prothrombin, and this is activated prothrombin that then makes that turn into thrombin. So let me get to the second part. Discuss the role of TPA, and so I'll actually use this one. TPA is the first thing that will start degrading the fibrin layer. So TPA is an activated form of, what is it, plasminogen, and then plasmin. Okay, so this will activate plasminogen, which turns into plasmin, which degrades fibrin. So then after the platelets, find an open area, start this process, get a whole bunch of them together, and then this one of these two pathways will start, and then reach to the common pathway, will then shoot out these fibrin strings like a net to pull everything in, closing up the opening to make sure everything's a strong mesh. The platelets will then contract to make sure everything's nice and tight. And then once that's all good and done, and things are healed up, TPA will be sent out that transfers, that activates the plasminogen into plasmin, which degrades the fibrin, so everything could be washed away.